Okay, so then today, what I would like to go over briefly, because of the poor video quality that's on the Canvas site, I'd like to go over chapter 7 and that little bit of 3, because I did grade your selective and differential medias, and let's just say I was less than impressed, right? So I have another similar lab activity to give you that's just like going to be like as a take-home homework assignment to help you kind of process that. So like I said, I'm just going to kind of run through this and try to hit important highlights of it so that it doesn't take us forever. All right. So the vocabulary that I put on there, listed out there, is literally just to kind of help you with all the words, because there is a lot of just, do you know, blah, the definition to blah, right? Many of these words you may have encountered before in some other biology class. Great, you just kind of know what they mean. Others of these words have very specific definitions that you kind of need to know for microbiology. Okay, so that's why I just left them all there. So because I think it helps you sometimes to kind of take a look through and see like where the important highlights are. So what I usually like to do for chapter 7 is to talk about nutritional requirements, because I think the chapter is called microbial nutrition, right, and growth. Right? And I think it's important for us to call it nutrition because they're a living thing. Right? They're cells and they need a full complement of all the raw materials. Unlike you, they can almost all make everything from just the raw materials, right? For a lot of parts, you can't do that. There are amino acids that you can't synthesize that you have to eat, right? There are all your proteins you make, most of them, right? So it is kind of important for us to start sort of at the very beginning. And we'll start with essential nutrients. And the first one, the biggest one, definitely is carbon, right? So in Bio 1, I usually do this, right? Because that's often the collection of essential nutrients that people discuss, and it also almost spells schnapps, so college students can remember that, right? So sulfur, carbon, hydrogen, nitrogen, oxygen, and phosphorus. I guess there's really no S at the end, right? We're going to start with carbon because you got to have a carbon source if you're going to build any organic materials, right? So let's start with why do cells need carbon? So what categories of molecules is carbon a part of, like specifically? Organic? Yes, organic molecules like? What if I do that? Carbohydrates, good. Right, like glucose, right? Lactose, starch, all those things. What other organic molecules do you build with carbon? Lipids, yes. And I'm just going to put in parentheses here or next to, right? Phospholipids. Super important because all membranes and all cells are made of phospholipids, right? And I'm also, it's not really how you spell lipids, is it? And I'm also going to put sterols here because in addition to lipids, pretty much most cells put some sort of sterol in their membrane, either cholesterol or other like ergosterol or fungus, different types of sterols. What else? So carbohydrates, lipids. What's the next one on the list? Proteins. And what are proteins made out of? amino acids. And you might remember from BIO1 that amino acids have their core is nitrogen carbon carboxyl group. Right. I think it's easy to forget that carbon is part of an amino acid because we focus on the amine group, we focus on the nitrogen. And what's the last one on this list? Right, nucleic acids, which remember are made out of nucleotides, which we spent a long time talking about before the last test, right? Those adenines, guanines, cytosine, thymines with their sugars and phosphates that make DNA and RNA, right? Those nucleotides, the purines and the pyrimidines, they are, we say, nitrogen containing rings, but it's not just nitrogen, right? It's nitrogen and carbon in those rings. What, you're just like, oh my God, why is she going over this again? Because I just graded your tests about replication, transcription, translation, right? So that's kind of on my mind about some of the gaps that we still have about how all this stuff fits together, right? So the nucleic acids are important because they hold the code to make all the proteins that do all the work in the cells, right? And the work they're doing is chemical work, chapter 8, building ATP, right? So that they can make more of these things and do other stuff. Okay, so cells need the carbon for all of these organic molecules, which we could have just said organic molecules, which is what I'm always tempted to do because I'm always tempted to think, oh, they know that list by now, right? But at the beginning, I think it's good to just hit it again. So where does the carbon come from? What are the major sources of carbon? Okay, carbon dioxide, carbon frequent. Oh, yes. So that brings us to that awful table, right? It's on page 173. So the sources of carbon. I can't spell today at all. Let's try that again. There's two, basically. Carbon dioxide, what we call inorganic carbon. Right? Because it's not part of an organic molecule already, and organic molecules. That in itself is what designates how this flow diagram or how that chart is pictured in every biology book, right? So I like to start with carbon source. Right, and divide it into those two. For me, it helps if it's not just lists of words, but if there's a picture now and then or some sort of a diagram, right? And if you use carbon dioxide as your carbon source, we call you an autotroph. And I think people often confuse that because they think only plants are autotrophs or only something that's photosynthetic is an autotroph, and that's not necessarily true. Because bacteria, above all else, all other kinds of organisms, there are bacteria that fit into each one of those characteristics, each one of those categories of nutrition, right? If an organism doesn't use carbon dioxide, if they actually use organic molecules as their carbon source, then we refer to them as a heterotroph. The words we put in front of autotroph and heterotroph are based on where the organism or how they make their energy. Okay, so the autotroph and heterotroph just have to do with where you get your carbon from. Then how you make your ATP. So we say energy source, but really what we're talking about is what kind of metabolism do you do? Aerobic, anaerobic, ferment? What kind of metabolism do you use to make your ATP? Right? Where do you get the energy to make your ATP? If you use light energy, sun, to make your ATP, right? Then we call you a photoautotroph. That's an easy one. And we're back to being a plant, right? I would remind in microbiology, it's not, we're not talking about plants, right? We would mostly be talking about algae here, right? And photosynthetic bacteria like cyanobacteria. Remember oscillatoria in BIO1 and spirogyra in BIO1, those things you look at under the microscope, Anabina. There was another one that looked kind of like that. Anyway, yeah, those would be cyanobacteria, right? Desmond, no, those are actually yeah, they're an algae. Those would count as an algae, yes. Like Desmus, right? The little Martian guy. One of my personal favorites. Now you can use carbon dioxide as your carbon source. Remember, plants and algae and cyanobacteria are going to take that CO2 plus the sunlight, and they're going to produce glucose, right? They're going to do photosynthesis of some sort, and then they're going to take those glucose and break them down to make ATP, right? If you use simple inorganic chemicals or inorganic molecules as your energy source, then we call you a chemoautotroph. Right? And there aren't any animals that are chemoautotrophs. There are only bacteria that are chemoautotrophs. And I think they give us some specific examples like the methanogens, which of course would belong to the archaea, and the rock-eating bacteria, some thiobacillus, right? So they use like silica and phosphates as their source to produce ATP in their electron transport chains, if you will. So I'll put methanogens down here. That's not how you spell that either. Sorry. There. Now, if you use organic molecules as your carbon source, again, then we can divide this up by how you make your ATP. Right? Believe it or not, there are organisms that can use sunlight and do a different kind of photosynthesis. They basically have photosystem 1 only, and it's a loop, not the electrons going on to photosystem 2. Or wait, they just have photosystem, yeah, they just have photosystem 1, not photosystem 2 in front of it. Then we would call you a photoheterotroph. And again, you don't know any of those, most likely, because there no animals are photoheterotrophs, and not plants either, right? Well, there might be a few, but mostly, again, bacteria are going to be photoheterotrophs. Let's see if they give us an example of one. Purple and green photosynthetic bacteria. So I'm going to put the purples on here because to remind us that you can use other colors, right? You can use other pigments to absorb the light and produce energy from it, right? And these guys are not going to be using carbon dioxide like plants do and like the cyanobacteria do. They're going to be using organic molecules as the basis to be able to produce their carbon, right? So they're organic, all of their other organic molecules, not carbon dioxide. And then last but not least, if you use organic molecules as your energy source, right, to make your ATP, so think aerobic respiration, right? or fermentation, where you're using glucose and you're breaking it down and using electron transport chain to make your ATP, or just simple phosphorylation, then we call you a chemoheterotroph. Right? Yay, now we have some animals, right? That would be you. You're a chemoheterotroph, right? All the fungus are chemoheterotrophs. Remember, they're a special kind of chemoheterotrophs. They're saprobes, right? And we would also throw all the parasites in this, all the animals that are parasites in as chemoheterotrophs, right? Because they're using organic material, you, right? They're living on you or some other animal as their source of energy and their source of carbon. So that one's probably the most involved because it's sort of the most important, right? Where do you get your carbon and how do you make your ATP? Those are like the two basic things. Like I've said, you've seen some version of this in other courses, especially like in a BIO 1 or a BIO 2, but most of the time we just talk about the photoautotrophs and the chemoheterotrophs, right? Because we're just talking about, we're talking about plants in chapter 9 and algae, and we're talking about animals for, aerobic respiration, right? Bacteria belong in every category, and new bacteria are found in all these categories in the environment all the time, right? They just don't happen to be the ones that cause disease. Try to remember back to the beginning of the semester when I told you there's only about 150 or so bacteria that actually cause disease in animals and humans. There are millions of species of bacteria, right? Okay. So carbon, I'm going to leave my list so I remember to cover all of it. Carbon, nitrogen, let's do nitrogen next. I think it's on my list, yeah. And I've left it blank there. What's the source, the main source of nitrogen for organisms on the planet? Gas, yes. Nitrogen gas, N2, right? 70% of the air in this room is nitrogen gas, and you can't use it. You can't use it. You can't use it for your to make ATP or an energy source, and you can't actually use it as a nitrogen source. Okay, all the nitrogen has to be converted to nitrates and nitrites. Those are usable forms of nitrogen. And guess what organisms on the planet do most of that converting from air, nitrogen gas, into nitrates and nitrites? If you had to guess, because you're in microbiology. It is bacteria, yes. So it is the prokaryotes that do most of this for us, particularly our friends cyanobacteria. So not only can they grab carbon dioxide and turn it into, and sunlight and turn it into ATP and produce oxygen for us, they also fix the nitrogen. And another big component are a lot of bacteria that are what we would just generically call soil bacteria, like Pseudomonas and Bacillus, right? Like thinking of bacteria that you've seen in the laboratory or touched in the laboratory, right? So the biggest source is gas, nitrogen gas. We can't use it unless the bacteria convert it into nitrates and nitrites for us. Even the plants have to have bacteria in their little nodules attached to their roots, right, in the mycorrhiza, in order to have these forms of nitrogen. Why do we need nitrogen? To make amino acids, to make proteins, yes. And where else do you find nitrogen? Where? Yes, in the nucleotides, yes. I thought you said that, but I couldn't. Friday, we all get to demask on campus. Yay! Right, permanently. Why, you ask? Because Friday is Designer Bag Bingo, a foundation event to raise money for scholarships for youth. And suddenly they couldn't put enough people at the tables. I shouldn't say that out loud. I was like, when are we, like the governor says we don't have more men. I'm like, no. Not until Friday. I'm like, oh, because that's bingo day. Okay, let's see if that's nitrogen. Great. Oxygen. Let's talk about oxygen. That's a big one. What is the main sources of oxygen? Air, right? Oxygen gas, O2, and drip. Water, yes. Right, water getting split via photosynthesis, right? To produce oxygen for you to breathe. The plants are doing that for you again, remember? Okay, what do you need oxygen for? What are enzymes made out of? Think about the element oxygen. Where do you find it? Proteins, where else? Carbohydrates, the O for oxygen, right? So carbohydrates, lipids, have glycerol in them, and then chains of fatty acids, steroids, right? Proteins, yes, and nucleic acids, right? So we need it for everything, right? So cells need it for everything. And yes, we need it for that's why you said ATP, sorry. You need it for a final electron acceptor, as on the electron transport chain, if the organism is doing aerobic respiration. Right, because the electrons from the NADH end up at oxygen, the protons end up fueling the ATP synthase to produce the ATP, right? Great. I don't know if I should talk about that other part right now or not. Where did it? See if I put that somewhere. No, okay. Hydrogen. So first and foremost, what are the sources of hydrogen? I'm not going to list all of them, but definitely water, right, on that list. Salts, yep. Hydrogen gas, like H2S, right? A lot of salts and acids have hydrogen in them, obviously. Why do we need nitrogen? There are 3 things listed there. I'm not going to rewrite all of them. I left them blank on your paper on purpose. So we need them for maintaining, actually I lie. We need them for maintaining pH, right? Being able to modify pH inside cells, that would be buffers. We need them for forming hydrogen bonds, which you might remember kind of hold all of your DNA together and hold water molecules to other water molecules, because water is polar covalent, blah, 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 right? And free energy. What does that mean? It means that proton pump, right, of ATP synthase, of the protons being pumped across the membrane. Yeah, chapter 8 never goes away. I'm sorry, ever. In all of biology, something always comes back to how you make ATP. It's like the central thing, right? ATP synthase requires, right, hydrogen ions in order to be able to produce the ATP. Phosphorus, put that on our list quickly. Main source of that is going to be phosphates, rocks, and dirt. Water dripping on rocks, basically, to produce phosphates and phosphoric acid. We need that, of course. Silt, I like to say, right? We need, think about every place that we have phosphates, right? So obviously ATP has phosphates in it, right? Phospholipids, all the nucleic acids, I mean, all the nucleotides have phosphates in them, right? So that's a lot of stuff. We don't need it in huge quantities, but it has to come from somewhere. Cells require need, okay. Sulfur, let's put that on the list. Sources, I believe, are also going to be rocks and sediments. And hydrogen sulfide gas. So H2S here, we can break that apart. Cells can break this apart, which certain bacteria can, of course. You're going to be able to produce hydrogen gas and sulfur, elemental sulfur, that's going to be turned into sulfate and sulfites. Why do we need this? Mostly for vitamins, disulfide bonds. So we don't talk about it a lot, but cysteine is one of the 20 amino acids that has a sulfur in it. And it is crucial for a lot of protein structure that there are cysteines that form a disulfide bond, a sulfur to sulfur bond in them. And if it's not produced, then the enzyme doesn't have the right shape and it can't function, right? So even though, you know, we don't think of it very often, not the same as we think of, oh, you have to have phosphorus or you have to have oxygen and carbon, it's certainly on the list of the most important. Micronutrients I put on there, right? So anything we need in small quantities for bacteria, like when we're trying to feed bacteria in the laboratory, the micronutrients often come from water. Like we use tap water, not distilled water, to make our media. And that's mostly because there's more junk in the tap water, right? There's more residual metal ions, there's more organic material. And so it's those micronutrients can be provided that way rather than having to concoct some sort of chemical slurry to add to the media, right? Micronutrients, growth factors. Yeah, those are kind of the mystery things. So right, so an essential nutrient is something you can't make, right? Just like an essential amino acid is one that you can't make. Of those micronutrients, right, we have those, and then we also have organic growth factors. Yeah, well, that usually means, again, vitamins. And it also usually means things we don't know the exact chemical composition of, like blood, right? So like we actually make chocolate agar and blood agar. We don't make it, but we buy blood agar and chocolate agar, which are both have components of blood in them, right? And so some bacteria will only grow if you feed them that, right? We call them very fastidious or picky bacteria. They have to have the right, not only macronutrients or essential nutrients, but all the right little tiny micronutrients and organic growth factors. And sometimes they are literally called like factor Y or factor X because you don't really know what they are, right? You just have to add this and then it will grow. Kind of like the rat liver extract thing with the Ames test, right? When I put that on the board and said, don't know why, but it makes it work better. So you put rat liver extract in and then the Ames test works, right? So sometimes there's things like that. All right, now, after we've talked about all of these things that you have to include in order to make bacteria grow, it would be a good time for us to talk about different types of media. And as I said, I graded many of your exercise 12 in lab, which has to do with selective and differential media. And it seems like there was still a little confusion about that. So now we're going to jump ahead to our little bit of chapter 3. And talk about media. So this was included in the discussion in chapter 3 because we consider it one of the part of the 5 I's, right? And in the beginning of the semester, I said we're just going to skip this part because we're not really ready to talk about how to feed the bacteria yet. We just need to get you to know a little bit about the bacteria before we start talking about how best to keep them in the laboratory, right? So physical states of media, you're very well familiar with this, right? It comes in the broth form, which is a liquid form, or we call it agar, which has a polysaccharide added to it. And that polysaccharide is purified from, believe it or not, some red algae. And the reason we use agar and not protein, although we're going to do a protein test today, we're going to do a gelatin test, we're going to set that up today and look at it on Thursday in the lab. The reason we use agar is because it is a solidifying agent. So it will make the media solid, so the bacteria don't grow in it, they grow on it. And it is all capitals, not a carbon source for prokaryotes. So they cannot digest it or break it apart like they can starch and use it to make energy or use it to as a building block to make more proteins, carbohydrates, nucleic acids, blah, blah, blah. This is key, not a carbon source. So any of you who wrote on that lab when it said, what is the purpose of the peptone? And you wrote, it's a source of protein. So it's a carbon source and a nitrogen source. That's true because peptone is partially digested protein, which has nitrogen and carbon in it. But then when you wrote agar is also a nutrifying, no, out came the red pen, right? Because does not, nobody eats it. Gelatin, which is protein based, some bacteria do eat that. They use it as a carbon source and they're going to turn the gelatin into liquid. So you've had Jell-O in your life somewhere, right? That is made with gelatin, spelled differently, right? But basically it's, you don't want to know where it comes from. But anyway, comes from bone marrow. Yeah, bones, yeah. So it's part of that use every part of the animal thing, right? And then it's purified, obviously. And then if you digest it, which you can, then that would not be a good, you've seen Jell-O makes things solid, right? But you wouldn't be good to feed that to agar because they would turn it into liquid. And then you wouldn't be able to identify them. You wouldn't be able to pick a colony off of the surface because it would turn it into a sort of sloppy broth, right? The other type of media that we've used, but you may not have really noticed, is what we call a semi-solid media. And that was like in the OF glucose tubes, where it's a less concentration of agar. So it's not completely solid, but it's semi-solid, right? So yeah, you've experienced all of those things, right? The other really cool thing about agar, not only is a solidifying agent and not only is it a carbon source, but because it's not protein, because it's polysaccharide, what happens to proteins when you heat them up over 60 degrees Celsius? What happens to an enzyme? Yeah, they denature. Okay, that'd be no good. If I had to heat it up to sterilize it, and then cool it down to feed it to the bacteria, or use it as it wouldn't go back to being solid, right? Because it is a polysaccharide, you can heat it like oil, 100 degrees Celsius and higher. So you can sterilize it in an autoclave, which is 121 degrees Celsius at 15 pounds pressure, right? And then you can cool it and it will form a solid again, which is kind of like a magic trick, because most things, anything that's protein based, it may form a solid or be gelatinous, right? But then when you heat it up to boiling and then cool it back down, probably not. And you can do it multiple times. Like, let's say, we haven't done this in class, but you might have agar that's been solidified in glass tubes that was autoclaved and solidified, but now you want to pour those into a petri dish. All you have to do is put the glass tubes that are sealed in a water bath, literally like boiling water, so a beaker with some boiling water in it. The agar will go back to liquid, and until it cools to 40 degrees Celsius, you can pour it wherever you want. You put it in a petri dish, you can put it in another tube, you can do whatever with it, and then it'll solidify when it gets cold again. So that's kind of cool. And I'm just going to put repeatedly, repeat, and still will solidify. So that's kind of another cool benefit of agar. Okay, so chemically defined media. That's usually also known as synthetic media, because we know every single thing that's in it, right? Not like peptone, right? That's partially digested proteins. That one we would call complex media. Like blood agar, things that have peptone in them, tryptone. Literally, those stand for partially digested proteins. Peptone is proteins that have been digested with pepsin. Tryptone is proteins that have been digested with trypsin. Nutrient agar, for example, would be that we use a lot in class. If you don't add the agar, it's nutrient broth. The agar just makes it solid. It's a complex media because it has peptone in it, right? And some salt, basically. Different kinds of salts that serve as a source of not only chloride ion and sodium, but of course, they can be used as sulfur. Okay, blood, blah, 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 blah. Okay, functional types, general purpose, right? We've talked about that before. Nutrient agar, TSA, which we're going to use a little bit coming up soon, stands for tryptase soy agar. Tryptase soy. Right. Those are sort of general use media because everybody will grow on them, or most bacteria will grow on them. They are not. So. General purpose, right? Everybody will grow. They are not like synthetic, selective or differential or enrichment media or anaerobic media. Those are like the big four types of sort of non-general purpose media. So let's start with enriched. Enrichment media is a tough one because it's kind of it's almost like a subcategory of selective media, in that it's where you add the use of it is to isolate bacteria that are in small numbers in a particular sample. So think environmental situations where you might collect 100 milliliters of water and there's only 4 bacteria in there that of the kind of bacteria that you want to study. Or you collect a big surface area of soil and there might only be 2 bacteria in there that you want to find. So what you do is you make enrichment media, you give them their very favorite food or the very thing that they must have to survive. And you're enriching the culture. They're going to grow a little bit better than they would if you just put them on nutrient agar. And if you enrich just for the bacteria, the special thing that they like most, a lot of times other bacteria don't eat that and don't grow as well. Right? So like I said, it's kind of like a selective media because you're really only selecting for something, but you're selecting for, this bacteria has to have asparagus with mustard sauce on it, or it will not grow, right? And most other organisms won't even look at the asparagus with mustard sauce. They can't even use it, right? So examples of things that you would really put in enrichment media are maybe a high concentration of phosphate or a high concentration of sulfate, or sometimes literally people will make a media and add some soil to the media because it's a chemical X. We don't really know what it is in the media that those bacteria like, but we know if we add some dirt, they'll grow in it, right? Selective media you're a little more familiar with because we've used it in class multiple times now. I'm going to erase all these guys. And it seemed like you guys did get a good kind of grasp on the selective media type. So selective media, it's enriching for or encourages growth of one type of bacteria and this is a big and inhibits, or we say selects against instead of selects for, right? Other types of bacteria. This is often a gram-positive, gram-negative thing. Right? So in the case of us using mannitol salt agar, for example, the high concentration of salt was selective. Staphylococcus lives on your skin. Your skin is salty. It likes that, right? Most gram-positives tolerate more salts than gram-negatives. So the salt selects for gram-positive organisms, right? And inhibits the gram-negatives because high salt concentration usually slows them down, E. coli, which normally would grow anywhere, right? Same for the EMB. And this is a part where, again, because you weren't super familiar with it, I get it. A lot of you picked one of, when it asked which of the media is selective and differential, you picked one of them. It turns out both of them are both selective and differential, right? So the EMB has bile salts in it, which means like ooze from a liver, right? And because E. coli is a enteric bacteria, because it lives in your digestive system, it's okay with that. It likes bile salts. Gram-positive organisms don't particularly like the bile salt part of it. Salt, yeah, not the bile part, right? So that inhibits the gram-positives and encourages the gram-negatives, particularly gram-negatives that live in your gut, that live in your digestive tract. The differential part is a little trickier because I'm going to give you an example of a media that is only differential, and then we're going to talk again about the mannitol salt and the EMB that are both selective and differential. Differential media is a little bit cloudier, I think, or harder for students to wrap their heads around, because differential media, many, let's say, or multiple bacteria will grow on it. But, this is a big but, you can distinguish between the bacteria that grow. You can tell the difference between them. You, meaning the lab person, can distinguish or differentiate between what grows. And probably the best example of this, the easiest example of this, is blood agar and streptococcus with a capital S. So the genus Streptococcus, capital S underlined, right? They all love blood agar. They'll all grow there. And inside your mouth, you have multiple different kinds of Streptococcus. You have Streptococcus mutans, Streptococcus mitis, Streptococcus salivarius, and occasionally you have Streptococcus pyogenes. Only Streptococcus pyogenes gives you a strep throat. The others do not. They're normal microbiota. You have them in your mouth in various concentrations all the time since the day you were born, right? But if we use blood agar, Streptococcus pyogenes and Streptococcus pyogenes only, as far as a pathogen of humans, digests the red blood cells that are in blood agar, and literally we call it, they have an enzyme that's called hemolysin, because it lyses red blood cells, right? And literally it turns clear. So blood agar is red, as you would guess. And when Streptococcus pyogenes is grown on blood agar, it lyses the red blood cells, and that part of the agar is not red anymore. It's clear. You can see through it. You can hold it up and see whatever's behind it. Only Streptococcus pyogenes does that. None of the other streptococci in your mouth do that. None of them. This is the only Streptococcus that is actually a pathogen of humans. Right? Streptococcus genus, right? It's the only one that's pathogen. So we have blood agar to test really quickly in a day if you actually have a strep throat or not, right? So I realized they do an instant test in the doctor's office. They can do a rapid test, which is based on antigen antibody binding, but is also a little bit more dependent on how the nurse practitioner, nurse physician, nurse's aide actually administers the test. Whereas this is the other reason that they not only do that test, but then stick the cotton swab way back in your throat, makes you cough, right? And they smear it on a blood agar plate, because if you have Streptococcus pyogenes, it will do this tomorrow. And then if they gave you the wrong antibiotic, they will give you a different one, right? Simple as that, right? It's also called Streptococcus pyogenes because it forms pus, right? So sometimes with a bright light and moving someone's tongue out of the way, you can actually see the pustules in the back of your throat. Now, super powerful for, think about the 5 eyes. Think about the last I is identification. Super powerful for identifying a bacteria really quickly is media that is both selective and differential. but you have to know, already kind of know who you're looking for, right? So sometimes this will bias the results that you find, right? So a person, let's say, has a particular infection that a physician suspects is E. coli or is salmonella. You will put the specimen you collect, the feces, the pus from the wound, whatever it is, you will put it on media that is selective and differential right away. When you're streaking for isolation, you'll streak on selective and differential media, probably like 3 of them, right? That are all for gram negatives. Let's say it's salmonella, right? All that are gram negatives. So one might be EMB, one might be McConkey, and then one might be one that specifically will grow salmonella only. So it's selective and differential just for salmonella. And then tomorrow you'll know if the person has one of those or all 3 of those. And you can then further test those organisms to see what they are. So selective and differential media does both. It selects for some, inhibits others. And like I said, that's usually a gram positive, gram negative thing. So our example of mannitol salt, right? The salt is the selecting agent. It's going to encourage gram positive bacteria to grow. And you can distinguish, differentiate, because we put mannitol and a pH indicator. Mannitol is a sugar. Only some gram positive bacteria will eat that sugar, produce an acid that will change the pH, which will change the media from red to yellow. Okay. So this is sugar, right? So some specific bacteria, like Staphylococcus aureus, for example, but not Staphylococcus epidermidis, will digest the mannitol, produce an acid, which will change the pH indicator from red to yellow. Remember, the plate turned yellow, media turned yellow behind the colonies. Some of you said the colonies were yellow. I was like, yeah, that's okay, whatever, you can call them that, right? But the colonies themselves didn't turn yellow, the media behind them turned yellow. This is very powerful. This is most of what the biochemical tests are based on. Think about the fermentation tubes that you set up. Glucose, lactose, and sucrose, remember? There's a green cap, yellow cap, brown cap, right, with the little germ tubes in them. I know, it was a long time ago. Over a week ago, it's hard to remember, right? And some of them turned yellow and some of them stayed purple, right? And some of them they turned yellow and made carbon dioxide because they were fermenting, right? So that media technically is also selective and maybe differential, depending on who you're testing. Because E. coli, Enterobacter, alkaligenes, and Pseudomonas did different things to those media. So you can use that to identify them as a biochemical test. There is a big book. It is called the Bergey's Manual of Determinative Bacteriology, because it's a nod to 1900. Right? Determinative, this is the clinical version, so we can determine who the bacteria is in the sample, right? The end of the 5 I's, the biochemical tests. And the Bergey's Manual of Determinative Bacteriology is a large book, and in it are many tables. So first it describes like all the gram-negative bacteria that you would find normal flora and cause diseases in human digestive tracts. And then for each of those bacteria, it will list all of its characteristics. Like for E. coli, for example, it will say E. coli. a gram-negative facultative anaerobic bacillus. Right? Think about how many of those words you know, what they mean now. And then there'll be a table that will have what all the biochemical tests and all the selective and differential media you can use to find E. coli, separate from Proteus or separate from Enterobacter, by doing certain combinations of selective and differential media. That's what you're going to do when I give you your unknown. You're going to follow a flow diagram that's going to take you through different biochemical tests to find out who you have. Right? Selective differential, reducing media. That's the last one on the list. That's for if you're going to do anaerobic bacteria, if you're going to try to grow anaerobic bacteria. So it technically is also kind of a selective media. Reducing. It has a compound in it. It's called sodium thioglycolate. This will, you can think of it this way. It will absorb free oxygen in the media and lower the concentration of oxygen in the media. This is for bacteria that do not like the presence of oxygen, or oxygen will kill them. Right? So we use this reducing media when we're trying to grow anaerobic bacteria. Anaerobes. Okay, so that's page 1, page 2, page 3. Ugh, I hate this part. All right, so I'm going to stop there. I think that's a good place to stop.